Good morning, students. Today we are going to deal with the last topic of class 8, first chapter, microorganisms. Today's topic is harmful effects of microorganism and food preservation technique. So before going into the topic, I would like to introduce all of you with a new keyword, that is pathogen. So pathogens are the microorganisms which causes human disease. Nearly most of the microorganisms are ineffective to human beings, but there are certain microorganisms which causes disease in humans. So all those microorganisms which causes human disease are called pathogen. Next, we are moving to our topic, that how these microorganisms are transmitted from one place to another. So first one is air. So air is a source by which many diseases are transmitted. Means when a diseased person sneezes or coughs, then the air or the droplets that come out from their mouth carries the microorganisms in them. And when that particular air enters into the body of a healthy human being, it is going to cause disease in the healthy person. So example includes the TB or tuberculosis, measles and flu. Next one is food and water. So as we all know that the food that is sold on stall or remain uncovered for some time, then it serves as a site for the microbial growth. And when that particular food material or water is consumed by a healthy person, so that particular microbe is going to enter into the body of healthy person and going to cause the disease. So example of that particular disease include that typhoid and cholera. Then third one is through diet contact. An example is chickenpox and ringworm. So as we all know that there is certain disease which are caused when we come in contact with a diseased person. So chickenpox and ringworm are the examples of that disease. Then fourth one is through insects. So insects that cause disease in human body or carries a disease to a healthy person is called vectors. So these diseases like malaria and dengue are caused due to the female Anopheles mosquito, which serves as the carrier for this disease. Then last one is through blood or body fluid. So example includes hepatitis B and AIDS. So these diseases are caused due to contaminated blood. When a particular human being is suffering from this disease, then the blood of that human being contains the microbes in them. So when these contaminated blood come in contact with the blood of a healthy person, then it is going to cause a disease in the healthy person also. Then next, we are moving to our last topic, that is the food preservation techniques. So as we all know that the food contains sugar or water in it. So these agents serve as the ready-made source for the microbes to grow in them. So as we as we keep the food, it serves as a site for their growth. And when we consume that food, it is going to cause disease in our body. So in order to prevent the growth of microbes in the food materials, we use certain techniques which are known as the food preservation technique. So here I am going to discuss about certain techniques which is used to prevent the food from getting spoiled or contaminated. So first one is heating and cooling. So from the very early, people used to heat their food source in order to prevent the growth of microbes in them. So heating causes the killing of microorganisms. It is generally used for milk. Then I am going to discuss a new keyword, that is UHT or ultra heat treatment. So it is a technique in which milk is heated at 135 degrees Celsius for 1 to 2 seconds in order to kill the microorganisms growing in them. Then next one is cooling. Cooling is a technique in which we cool the vegetables or fruits at 5 degrees Celsius in order to stop or inhibit the growth of microbes in them. As we all know that the fruit or vegetables serves as the site for the microbial growth because it contains glucose or sugar in them. So in order to prevent their spoilage, we store them at 5 degrees Celsius. Then third one is sugar, adding sugar or salt. So adding sugar or salt to food materials causes the osmosis of microbes in them present in the food material. So osmosis is a technique in which the water from higher concentration moves to lower concentration. So adding sugar and salt to food materials causes osmosis of microbes present in them. Then third one is using chemical preservative. So chemical preservatives are chemical synthesized substance which are going to kill or inhibit the growth of microorganisms in the food materials. Example includes sodium benzoate and Ajinomoto salts, which are used in the packaged food materials like chip wrappers, etc. Fourth one is canning and packaging. So canning and packaging of food materials is done in order to remove the air from the particular food materials and stop the growth of microorganisms in them. So as we all know that if there is no air, then no microorganism is going to survive in them. So if there is no air, then the microorganisms will not grow, and it is one of the great techniques used in present days. Last one is pasteurization. So it is introduced by Louis Pasteur, in which it is technique in which a particular liquid is heated at 72 degrees Celsius for 15 minutes in order to kill the microorganisms in them, and is generally used for milk. So this ends today's topic. We'll meet in the next video. Thank you.